Excitable tissues, that is neurons and muscle, are characterized by their ability to respond to a stimulus by rapidly generating and then propagating electrical signals. That propagated electrical signal is the action potential. This is a constant electrical signal that we can propagate over potentially a long distance without any decay. In these sessions, we will define action potentials, talk about how they arise, what their ionic basis is. We'll talk about the importance of refractoriness and factors that are important for the conduction of action potentials. And then we'll finish up introducing the basic components of a chemical synapse as the main means of communication between excitable cells. Action potentials are an all or none impulse. They either happen or they don't. And they occur when an excitable cell is depolarized beyond a threshold voltage. Once the threshold has been exceeded, there is a phase of rapid depolarization, which then ends abruptly at a peak voltage. We sometimes talk about the overshoot, which is just the amount by which the peak voltage exceeds 0 millivolts. There's then usually a slower repolarizing phase that returns the membrane potential towards its resting potential Vm. In neurons, we also see something called an after hyperpolarization or an undershoot, in which the membrane potential is transiently more negative than the resting membrane potential. The phases of an action potential are explained by changes in membrane sodium and potassium conductance with time. Rapid depolarization after threshold voltage is exceeded is due to the opening of voltage-gated sodium channels. You see here on the graph G sodium, the sodium conductance is increasing. The peak voltage where rapid depolarization abruptly ends and the membrane potential enters its repolarizing phase has two components. The first is closure of inactivation gates in sodium channels. And so these are remarkable channels that are both activated rapidly by a threshold depolarization, but then with a sustained depolarization can also be inactivated. And that occurs because an inactivation particle swings into the pore of the channel. The second component, as we turn the corner into repolarization, is opening of voltage-gated potassium channels. And so repolarization of the membrane will now proceed with a decreasing sodium conductance and an increasing potassium conductance that you can see here. The green graph sodium conductance is decreasing, downswing here, as the potassium conductance here, shown in orange, is increasing. And because of that, we move away from E sodium and down back towards E potassium. After hyperpolarization occurs in neurons because potassium conductance is transiently even higher now than it is at rest, which causes Vm here to nearly approach the equilibrium potential for potassium. And then a millisecond or so later, all of the voltage gates and inactivation gates are reset, and we can return to the resting membrane potential. In the nervous system, a stimulus intensity, for example, the loudness of a sound, is encoded in the nervous system by action potential frequency. And that's because all action potentials have the same amplitude, and action potentials can never summate together. So it's not the size of the voltage change, but the frequency of the action potentials that encodes intensity. The maximum action potential frequency then is limited because a finite period of time must elapse after one action potential before a second one can be triggered. And this is what we refer to as refractoriness. There is an absolute refractory period, shown here in the red shaded area, which is the time from the beginning of one action potential when it is simply impossible to stimulate another one. This results from the closure of those inactivation gates in sodium channels, and these inactivated channels must then fully close. The gates must be reset then before the channel could be reopened for a reactivation event. There is then a relative refractory period, this blue shaded area, which is the time after the absolute refractory period when another impulse could be evoked, but only if there is a much stronger stimulus applied. Action potentials can only be propagated in one direction along a nerve axon or muscle fiber. The way this works is kind of analogous to those domino toppling experiments where once you topple the first domino, one falls into the next and to the next, and the whole string of dominoes will fall. So an impulse in one area here causes a local flow of current which depolarizes the adjacent area to threshold. That will generate then a new action potential downstream, and this process simply continues to provide a unidirectional current flow. The reason that this flow of action potentials is unidirectional is because the upstream region is still in its refractory period. The speed of an action potential conduction is faster in large diameter fibers because they have lower electrical resistance than small fibers, and this local current flow here moves very quickly in a large fiber. But as we're thinking about conduction speed, of course, of critical importance is the presence or absence of myelination. Myelin consists of glial cell membranes which are concentrically wrapped around the nerve fiber to provide insulation. In peripheral nerves, the myelin sheath is interrupted at regular intervals called the nodes of Ranvier. And what happens here is that the action potential propagation occurs from node to node rather than having to conduct along the entire nerve cell membrane. And that's because the voltage-gated sodium channel expression is clustered here in the unmyelinated node of Ranvier. And this is a process that is then called saltatory conduction, as the voltage bounces from one node to the next. You should be aware that there are important diseases of demyelination that can either affect the central nervous system, that's the syndrome of multiple sclerosis, or there are versions of this that can affect the peripheral nervous system, such as Guillain-Barré syndrome. And these conditions significantly impede nerve conduction and can have major impact on the function of the nervous system. So how is the information encoded by an action potential passed between cells? That, of course, occurs at synapses, which are these specialized cell-to-cell contacts that allow for this information transfer between cells. Synapses occur at the junction between the processes of two neurons, or between a neuron and an effector cell, such as a muscle or a gland. And in general, there are two types of synapse. There are electrical synapses that occur when two cells are joined by gap junctions, and this allows current flow from cell to cell through non-selective ion pores. A great example is cardiac muscle, and we'll see that's critical for the coordinated spread of electrical excitation and contraction in heart muscle. More generally, we're thinking about chemical synapses, which involve the release of a chemical transmitter by one cell that acts upon another cell. Action potentials in a presynaptic cell cause the release of a chemical transmitter. This will cross a narrow cleft to interact with specific receptors then on a postsynaptic cell. If excitatory neurotransmitters are released, these will depolarize the postsynaptic cell and produce an excitatory postsynaptic potential, or EPSP. On the other hand, transmitters can also be inhibitory, and these will hyperpolarize the cell, producing an inhibitory postsynaptic potential, or IPSP. In the Neuroscience Chapter 2, we'll talk more about how these potentials interact to form integrated neuronal circuits. So looking in a bit more detail then, presynaptic terminals contain neurotransmitter chemicals typically stored in presynaptic vesicles, and action potentials in the presynaptic terminal trigger voltage-gated calcium channels and are responsible for inducing calcium entry into the nerve terminal, which then triggers release of neurotransmitter by exocytosis. There's always then some delay between the arrival of an action potential in the presynaptic terminal and the onset of a measurable response in the postsynaptic cell. The delay can be very short, maybe a millisecond or so, and this will occur when the postsynaptic receptor is a ligand-gated ion channel. Many examples of that will come across, and in general we call these ionotropic receptors. A good example of that we'll see very shortly is in the neuromuscular junction. Alternatively, the delay can be a good bit longer, over 100 milliseconds, and that's if the receptor type is linked to a second messenger cascade. And in general, we talk about metabotropic receptors where this is the case. Finally, the transmitter action must be rapidly terminated. And in general, there are one of three processes that can remove a transmitter from the synaptic cleft. It could either simply diffuse away, It can be degraded by an enzyme, which happens to be particularly important for acetylcholine, the acetylcholine esterases. More commonly, the transmitter can be taken up either into the nerve ending or into another cell, such as a glial cell. In the next block of cell and general sessions, we'll start talking about muscle.